Hello students, I am Mrs. Shubham Patil. Welcome you all to study Introduction to Volumetric Analysis. In this video, we'll get introduced to the technique of volumetric analysis. Just imagine a situation in which a tanker carrying concentrated hydrochloric acid came across an accident. All the acid flowed to the nearby pond, making the pond water acidic. Imagine the extent of damage caused. By means of some quantitative analysis, we can find concentration of acid in the pond. So let's see about quantitative analysis. Quantitative analysis can be carried out in two ways: gravimetric analysis and volumetric analysis. Volumetric analysis is a method to determine the amount of substances in a given sample. It involves measurement of volume of solution of reacting substances. A procedure called titration is used in volumetric analysis. In titration, standard solution is added to a measured volume of an unknown solution until the reaction is completed. Students, let's have a glimpse at the required apparatus. Burette. This is burette. Burette is the laboratory apparatus used in quantitative chemical analysis to accurately measure the volume of solution. Burette is a long cylindrical tube of uniform diameter. It has a stopcock like this, or a turning plug as shown in this image, or spigot at the lower end, which is shown in this last image. Let's see what precautions to take while using burette. Check that the burette you are using is calibrated. Also see for the visibility of the calibration markings on the burette. Check that the tip of the burette is intact. Pipette. This is pipette. Pipette is the apparatus used for accurate measurement of definite volume of solution. It consists of a long narrow tube with cylindrical bulb in the middle and a jet at its lower end. On the upper part of the stem, there is an each circular mark. On the bulb is marked the volume which the pipette can deliver when filled up to the circular mark. So you can see, students, in this image, it is 10 written on the bulb. That means up to the mark, if we are taking the solution, that much solution will be of the measure 10 ml. Inspect the pipette to make sure that the tip is not chipped and the top end of the pipette is smooth and flat. If the top end of the pipette is chipped or not smooth, it is very difficult to control the flow of the solution from the pipette. Conical flask. Conical flask has a conical body with a narrow neck and flat bottom. This shape allows the flask to shake or stir the flask without spilling liquid. It is used to manipulate solutions or to carry titrations. These are conical flasks of various shapes. It's also known as Erlenmeyer flask, named after the German chemist Emil Erlenmeyer, who invented it in 1860. Beakers. Beakers are the cylindrical containers with flat bottom. They are used for mixing, stirring and heating chemicals. They are available in various sizes. Beaker has a wide mouth for pouring the solution easily. It has a spout or beak. You can see in this image a beak over here on its rim to aid in pouring. We use beakers in volumetric analysis for preparation of standard solution as well as for filling the solution in burette. Funnel. A funnel is a tube or pipe that is wide at the top and narrow at the bottom. It is used for guiding liquid into a small opening. Burette stand. You can see the images of burette stand here. Burette stand is a vertical stand used for holding a burette in upright position. Here you can see the assembly, how the burette is fitted on the burette stand and how the conical flask is placed under the burette. Burette clamp. It is used to hold and secure the burette on the burette stand. White tile. A white tile is placed underneath the flask so that any color change can be clearly observed against white background. Now we'll study for some important terms used in volumetric analysis. Standard solution. It is a solution which contains a known concentration of an element or a substance. The solution whose concentration is accurately known is called a standard solution. Molarity. It represents the number of moles of solute per liter of the solution. The solution which contains 1 gram molecular weight or 1 mole of the solute in 1 liter of its solution is called a molar solution. So molarity is equal to number of gram moles of solute divided by volume of solution in liter. Thus, molarity is equal to mass of the solute in grams upon molar mass into volume of solution in liter. 1 molar solution means 1 mole of solute per liter of solution. Strength. Amount of the substance in grams dissolved per liter of solution is the strength of that solution, and it is equal to molarity into molar mass of solute. Molar mass. It is an algebraic sum of atomic weights of all the atoms present in the molecule. Titration. The process of carrying out a volumetric analysis by using a suitable standard solution, volumetric apparatus and indicator is called titration, or it is a process involving a chemical reaction between a standard solution and the other solution whose concentration is to be determined. Types of titration. Depending upon the nature of reacting species, titration methods are generally classified as follows: acidimetry or alkalimetry, oxidation reduction titration, that is redox titration, iodometric titration, and precipitation titration. Titrant is the solution filled in the burette. Titrant is the solution taken in the conical flask. End point. It is the point when the reaction between two solutions in a titration has just completed. The end point is indicated by change in color of the reaction mixture during the reaction. Equivalence point. It is the point when the reaction between two solutions has been completed just before color change. Indicator A substance used in titration to indicate the completion of a reaction, that is the end point, by its change in the color is known as an indicator. The choice of indicator depends upon the nature of acid, base or the substance involved in the reaction. So let's see how the indicator is selected. If acid is strong and base also is strong, we can use indicators like phenolphthalein, methyl orange or methyl red, which gives us the color change once the reaction is completed. Phenolphthalein changes its color. In acid it remains colorless, but in base it becomes pink. Methyl orange has pink color in acid, whereas it has yellow color in base. Methyl red indicator is red in acid solution, but in basic solution it changes its color to yellow. If our acid is strong acid and base is weak base, the indicator used is methyl orange, which stays pink in acid but becomes yellow in base. If the acid used for acid-base titration is weak acid and the base is strong base, the indicator of choice is phenolphthalein, which is colorless in acidic medium but turns pink in basic medium. So this is the way how we can select the indicator for our titration so as to know about change in the color which indicates completion of the reaction. So this is all about the introduction to the apparatus and the terms involved in the volumetric analysis. Thank you so much for your listening.